Hola bio. Okay, so last class period, you took down notes on this process of cellular respiration, and wait 1 second, I'm going to bend it right now. Okay, here is the note, and boop, boop. Okay, so cellular respiration, as you know, it can happen in the presence of oxygen and also in the absence of oxygen. And we're going to be focusing on the aerobic pathways in a few screencasts from now. And today's screencast is going to be about the anaerobic pathway. So just focusing in on, I can't zoom. Just focusing on this section here. So I'm going to get a new page, and here we go. Okay, so anaerobic respiration starts out in the cytoplasm, and much like, oh, that's terrible. Okay, starts out in the cytoplasm with a molecule of glucose. So this is one glucose molecule right here. And remember, it's a 6-carbon molecule. And this molecule of glucose is going to enter glycolysis. And we're going to depict glycolysis as a little box. So let's write it down here. And give yourself a big empty box here. Okay, so our glucose molecule enters glycolysis, which again is taking place in the cytoplasm. Oh, I want to copy that, not move it. So copy your glucose molecule. Oh, no, damn it. I mean, darn it. And paste it right in this box. So your glucose molecule enters glycolysis. And then off to the side, I want you to write whoa, not that. Just flip back to the wrong page. Okay, off to the side, write 2 A T and then P, but you're going to draw the P in a funny way. Okay, so a little P sub I there. So this is indicating that there's phosphate group. There's actually 3 phosphate groups on this ATP molecule, and we have 2 of them. Okay, so your setup looks like this. You've got your glucose at the top and your ATP on the side, and so the first step of glycolysis is just entering glycolysis. So let's put a little 1 next to glycolysis here. So first step, enter glycolysis. The second step is on this side. This is called the investment phase. And it's called the investment phase because we're actually going to invest 2 ATP in getting this process started. So, looks something like this. The 2 ATP, they're going to come in to our box, and they're going to drop 2 phosphate groups. So, what you're going to need to do here again is copy this glucose molecule and paste it down below. But on it, you are going to add your phosphate groups. Copy that. And paste a phosphate on either side. And the reason for this is that this phosphate group energizes this glucose molecule and makes it ready, to have work done on it. Or I guess a better way of saying that is it makes it ready to do work. And what's going to happen is, actually, I'm not even done. So the 2 ATP become 2 ADP. So what's actually going to happen to this molecule is after the phosphate groups have been added to it, it's going to split into 2 3-carbon molecules. This is called G3P. So I'm going to write that all down below. Okay, G3P. And in addition to these two phosphates that are now on either end, as you can see here, we also have some more free phosphate come into the mix. So the third step is more free phosphate, just from the cytoplasm, coming in and binding to these G3P molecules. So you have this free phosphate, it comes in, and then we have some more phosphate. But this is a free lunch. It doesn't take any energy to bind these phosphates to this 3-carbon molecule. But in the process of binding the phosphate to G3P, another reaction also happens. And I'm going to put this on the other side of the box. So remember from photosynthesis, we had our NADP+ molecule, and that was a high energy electron carrier molecule. You can think of a high energy electron like a piece of radioactive material. It has a lot of energy, and that can be used to do useful things, but it's also dangerous. You wouldn't want to just hold radioactive material, a lot of radioactive material in your hand, because it would cause damage to, most importantly, to your DNA. Well, a high energy electron is very similar in that it can also cause damage to your DNA and also to other things in your body, like your proteins. But the DNA damage is probably the most important and scary thing that your body wants to avoid. And so it can't just release free high energy electrons into the cytoplasm, because they're going to go off and they're going to wreak havoc. So instead, you have to carry them. So just like you would carry a piece of radioactive material in a lead box or a lead briefcase, your NADP+ in photosynthesis is like your lead briefcase. It carries that electron, it keeps it safe until it can be brought to the place where it's going to be used, and that was in the Calvin cycle in photosynthesis. But in cellular respiration, we have a slightly different molecule, but same general or overall concept applies here. However, this molecule is called NAD+. So you have two NAD pluses that come in, and they pick up two things from these G3P molecules. I'm going to move this. They pick up hydrogen, ions, and they pick up high energy electrons. So what pops out is two NADHs, which are now energized. They hold these high energy electrons. So this is also part of step 3. And as you probably, or most certainly remember from chemistry, if you lose an electron, you are oxidized. Leo goes Gur. If you gain an electron, you are reduced. So this molecule is reduced. This is a reduction, because it has gained an electron. The NAD+ gained an electron. However, this molecule over here, this G3P, is oxidized. Because it lost an electron. Oh no. So this is also part of step 3. So, 3 pieces. Number 1, the phosphate gets dumped off. Number 2, and 3, is that we have this reduction-oxidation reaction. So G3P is oxidized because it loses an electron, and NADH is reduced because it gains an electron. Okay, and this step here is the key. Okay, put my AP Art skills to the test here. So, oh, this is going to be a terrible key. This. Oh, that's so bad. Okay, it's okay. Next year, I'll get the key better. Okay, this is the key, this reduction, because it prepares this G3P molecule for what's called the payoff phase. And you can imagine if the investment phase we put in ATP, the payoff phase we're going to get out some ATP. So here's what happens next. We're going to take 4 ADP, 4 of these guys, and throw them into our reaction and strip away the phosphates from... Why did this carbon thing go away? That's better. Okay, strip away the phosphate groups on either side of our G3P and create more ATP. Okay, so this is step 4. We just generated 4 ATP. And we're left with 2 molecules, 2 3-carbon molecules called pyruvate. So again, 4 ATP, this is called the payoff phase. So our ADP come in and are charged up with this supercharged G3P molecule and become 4 ATP. And this is a pretty important process. Oh God, why is he doing that? And it's called substrate level phosphorylation. And substrate phosphorylation is different from electron transport phosphorylation in that the ATP, or the ADP, comes into direct contact with this molecule right here. So the ADP is actually stripping off a phosphate group directly from this molecule. So it's substrate level phosphorylation because this molecule is the substrate, as opposed to oxidative phosphorylation, which we'll talk about more later on in the process. Okay, but ultimately, out of our box, we get this guy, which is pyruvate, and there are 2 of them, 2 pyruvate. So what went into our box? 1 glucose. What came out? 2 pyruvate. Now, because this is a pathway of anaerobic respiration, there's no oxygen, and it can go in one of 2 directions, depending on what type of organism it is. And they're both going to be a fermentation pathway. Oh shoot, I am really late for community lunch. Okay, to be continued. Okay, I'm back from community lunch. It was delicious, but my table is the worst. No one talks. Okay, I totally forget where I was, but let me look. I think I was talking about fermentation. Okay, so there are 2 different types of fermentation. And the first one we're going to talk about is lactic acid fermentation. So just make a little arrow over here. Okay, so we're in a new section, fermentation. And the whole goal of fermentation is to regenerate our key, NAD+. Because we used it, we generated NADH, and so we need free NAD+ to run this reaction again and get these 4 ATP, which I'm assuming you did math here, because remember we spent 2 ATP at the beginning, this was our investment, and we got 4 out at the end, so in sum, our net total was only 2 ATP. But we can't even run this reaction at all if we don't have our key, our 2 NAD pluses here. So the goal is to regenerate this NADH back into NAD+. That's the overall goal of our fermentation process. So let's talk about how we do this in humans. So we start out with pyruvate, 2 pyruvate. Copy here, paste it right here, but I'm gonna 2 pyruvate. And once again, we have another box. And it's not centered. Okay, better. So these pyruvates enter this box. And paste. Paste. And I'm going to really simplify this process. It's actually much more complicated than I'm going to draw it here, but for you guys, we'll keep it simple. So, our goal is to regenerate our NADH back into NAD+. So we have NADH right here, and once again, there are two of them. And let's draw that in red. So 2 NADH. They're going to come in and drop off the exact same two things that they picked up before. They drop off hydrogens and high energy electrons, and this pyruvate is reduced. And this NADH is oxidized. Remember, if you lose electrons, you're oxidized. If you gain electrons, you're reduced. And out pops 2 NAD+. So now we've freed up this NAD+ to jump into this reaction again. And in reducing our pyruvate molecule, we actually change it, chemically change it, so now it is lactic acid. Okay, so once again, in this process, we have reduced our pyruvates and made lactic acid. But this is not the only way to do fermentation, and other organisms, for example yeast, do it slightly differently. They make, ultimately, ethanol. And let's just talk really quickly about how that happens. So the inputs are the same. So you can just take this, copy it, and paste that over here. So 2 pyruvates are going to go into fermentation. But this is alcoholic fermentation. So let's draw a box slightly differently. Draw this box like a little hourglass shape, because alcoholic fermentation actually has 2 pieces. Okay, so your box should look like this. And grab your carbons, 2 pyruvate, enter this right here. But before they move on, 2 waters, 2 H2O, come in, and they're in the form of a liquid here. So I'm going to do a small L. And out from this carbon, the 3-carbon compound, out pops 2 CO2. So let's just grab one of these, copy, and paste. So a carbon from each pyruvate pops off. So now we did have a 3-carbon compound, but now we're left with a 2-carbon compound. Here we go, 2-carbon compound, 2 of them. And this is called acetaldehyde. And it's this acetaldehyde that is going to be reduced. So our NADH is going to come in here, once again, drop off H plus and electrons, and become NAD plus. So this is oxidized. Oops. Okay, and in this oxidation reduction reaction, once again we chemically change. I'm just gonna cheat here, grab these, copy. We change the chemical structure when we reduce acetaldehyde, and out pops ethanol. Ethanol or alcohol. So 2 ethanols are the final result of this process. Okay, so here it all is. Oh man, I wish you could see it on one screen when it's too big. Damn it. Okay, well, here it is. Glycolysis followed by the 2 different fermentation pathways. Remember, this is done by yeast and bacteria, and this type is done by everybody else, including you. And overall, this whole thing is anaerobic respiration, the sum of glycolysis and fermentation. Okay, this screencast is way too long. Done.